Thank you very much to the organizers and to the people who made this talk possible. So I'd like to start by providing some additional motivation for this field, which is this curve. We've all heard about bending the curve in the healthcare field. This is the curve. So currently, we're spending around $17 out of every 100 developed in wealth within this country in healthcare. And if you extrapolate this curve out to the year 2050, 1 out of every $2 of wealth created in the country is going to be turned right around and invested in maintaining the health care of the people who are creating that wealth. Obviously, that curve is not sustainable. The bank is going to get broken somewhere along the way. So furthermore, the United States invests more per capita in healthcare than any other country around the world, and perhaps a little bit more sobering than that, if we pay attention to one country, which is a neighbor to the south, Cuba, we spend 20-fold as much money on healthcare per capita as they do, yet we have the same lifespan. So there are a number of potential reasons for this, and the majority of that growth in the health care cost within this country is due to this problem, that the country has evolved from situations where acute health care is the issue into situations where long-term chronic health care diseases end up dominating our healthcare field. So for example, neurodegenerative diseases, cancer, obesity, and obesity-related conditions such as type 2 diabetes and cardiovascular disease. It's estimated that if you add together the number of diagnosed as well as undiagnosed cases of type 2 diabetes, it's 28 million already within the country. Projected by the year 2050, there are going to be 10 million people with Alzheimer's disease. So unfortunately, although the fact that the incidence of these disorders is increasing, currently the treatment options to be able to address them are not. So currently, these are treatments that simply alleviate some of the symptoms for these disorders, don't treat the underlying causes necessarily, and therefore these treatments are long-term and chronic in nature and are quite expensive. So if you take a look at what these treatments look like, so the pharmaceutical field, of course, has been based upon small molecules over the past century. The first of these was synthesized in Germany around 1908, rational synthesis of antibiotics. And of course, small molecules have diversified in many ways and still form the majority of the healthcare options within the country. Protein therapies were developed not long thereafter with the Nobel Prize-winning discovery of a small crystalline substance known as insulin, which is missing in type 1 diabetes patients, and was this began as a treatment to treat type 1 diabetes back in the 20s. And over the past 10 years, of course, things like monoclonal antibodies have arisen as major healthcare products within the country, expensive healthcare products, incidentally. So gene therapy was conceived at least as an idea back in the 1960s, and over the past 5 years, the idea of using DNA as a medicine has begun to succeed clinically. And finally, I'm going to be discussing stem cells. So stem cells are the reason that bone marrow transplants work. So they've already been successful within the clinic. And over the past 10 to 15 years, people have begun to investigate their properties for the treatment of other tissues outside of the blood system. So there's a natural progression in these molecular medicines. Small molecules function by binding to proteins and changing their activities in a therapeutic way. Well, why not just deliver the protein itself, the gene encoding the protein, or the entire human genome in the form of a cell? And furthermore, small molecules and protein therapies, which are, as I mentioned, quite expensive, are temporary residents of the body. They have a finite lifetime, and they're turned over relatively rapidly, whereas DNA and cells, at least in principle, have the potential to be permanent components of a diseased tissue, and therefore can offer at least the long-term potential of a permanent cure. So it's these generations of therapies that I believe or hope are the future and could potentially make healthcare more affordable as well as improving significantly the quality of life of people who receive these treatments. So let's talk a little bit about what a stem cell is. You're all going to be very familiar with this material, but I just want to define a couple of terms. So if we start out with a fertilized egg, it divides in 2 to the nth fashion until you end up with a blastocyst stage embryo. The first in vitro fertilization baby just turned 33 years old, so IVF has been going on in this country for a number of decades. And as a result, the leftovers of IVF have been frozen down across the country. There are around 400,000 embryos frozen within the US alone, several hundred thousand more within Europe. So some of these would-be parents are now in their 50s and 60s. They've decided they're done having kids. They're willing to donate these embryos to science. And if you isolate the so-called inner cell mass out of this blastocyst, you can generate a human embryonic stem cell, which is pluripotent and therefore, this term means has the capacity to turn into every single cell type within the adult body. These cells, in turn, can differentiate and create tissue-specific stem cells, including the mesenchymal stem cells that Helen referred to, that make cells within the connective tissue. And neural stem cells, for example, that can create all the major cell types within the adult nervous system through complex processes that are denoted by these dotted lines. So in addition, all of us have pockets or niches of stem cells distributed in the organs throughout our body. Regardless of where they come from, stem cells share two defining properties: the capacity to divide in an immature state and make more of themselves, as well as the ability to move from the left to the right of the screen to specialize or differentiate into the functional cell types that make up adult organs and tissues. So there are a number of ways in which one can conceive of using these cells therapeutically. One approach is simply to manipulate their activities in situ as they exist already inside the body. And another approach is to isolate or derive these cells, expand them in number outside the body, differentiate them and assemble them into functional tissue equivalents, as was elegantly described in the previous talk. And as is going to be discussed in the next talk, these human tissue equivalents can be potentially used as the basis for high-throughput screening to identify better drugs, conventional drugs, to treat human disease. And in the longer term, one can envision taking these tissue equivalents, implanting them into the site of disease or injury in order to repair organs. So outside of the blood system, where this process is already working to an extent, there are a number of challenges or issues with implementation for other tissues. I'm not going to go through this detailed list. I'll just highlight a couple of the major concerns within the field currently. So there's poor control over cell culture systems. It's difficult to expand these cells while maintaining them in a sufficiently immature state. Once you decide to differentiate them, the complex and lengthy processes necessary to take this immature cell and create or mass produce an important cell type for therapeutic implications, these end up resulting in very heterogeneous cell cultures that contain many different cell types rather than the one that's of therapeutic interest. And finally, upon implantation of these cells in vivo into a tissue or organ, typically integration or engraftment is very poor. So, for example, in the central nervous system, less than 1% of the cells that are implanted often survive and can functionally contribute to the tissue. So why is it then that while biology, your bodies actually control each one of these processes in a very precise way on a daily basis with your own stem cells, why do we have such poor control over these processes outside the body? And what can we learn from biology that can teach us how better to develop cell processes to mass produce these cells? So essentially, we must learn best how to control, or better how to control these processes of self-renewal and differentiation for a stem cell. So if you look at a lower organism, so this is C. elegans, a worm or a nematode, and the development of this organism as a model system to understand how organisms develop in general was a Nobel Prize-winning work that led to a Nobel Prize in 2002 to Sidney Brenner. And this organism is so simple, in fact, that it's deterministic in its development. There are precisely 959 cells in every single adult nematode, at least the hermaphrodite form. And furthermore, the fate and the source of every single cell within this organism is actually very well known. So if you want to create this cell type here, well, you start there, you turn left, you turn right, and proceed through these various differentiation steps to be able to create a particular cell type, for example, the sensory neuron. So mammalian development, as you might not be surprised, is much more complex than this. But can we learn actually some principles from these hierarchies or these developmental pathways and apply that towards the creation of culture systems that can control analogous fate decisions for mammalian cells? So what is it that controls these fate decisions? Well, it's a combination of cell intrinsic factors, things that the DNA inside the cell tells it to do, combined together with extrinsic factors or information from the outside of the cell, biochemical molecules known as ligands. And there are receptors on the cell surface that serve as antennae to convey that information to the interior of the cell. This, in turn, unleashes the activation of very complex nonlinear biochemical reaction networks, signaling networks, as well as genetic networks. And these serve as nothing more than computational devices that enable a cell to sense the information outside of it and make decisions accordingly. So, for example, our goal then, if we would like the cell to have a particular behavioral output, such as divide for a while and then navigate down this differentiation pathway, our job is then to identify the set of signaling inputs that must be provided to the cell as a function over time. So these signaling molecules that are on the outside of the cell, and many of them were already very elegantly referred to in the prior talk, include a set of molecules that are usual suspects within the biology field, small molecules that float around, small proteins that either float around or are sometimes immobilized onto the solid phase. a cage or a network of proteins, the extracellular matrix, that surrounds every single cell inside the body and is responsible in large part for the mechanical integrity of our tissue. And finally, neighboring cells that are specialized and surround the stem cell, feed it with regulatory information. So our job then is to learn the relationship between these complex inputs and then the cell behavior as a function of those inputs. Now, these molecules are not simply floating around randomly in solution. We're solid phase organisms after all. And if you examine a typical microenvironment for a stem cell, this happens to be a stem cell that resides within a region of your forebrain inside your adult central nervous system, these have a lot of structure to them. So shown here in blue are the stem cells. They're contacting one another. They're contacting differentiated cells around them, such as ependymal cells, neuroblasts, neurons. They're contacting the vasculature, the endothelial cells that make up blood vessels, and they also are coming into contact with fluids, such as the cerebral spinal fluid that circulates throughout your central nervous system, as well as blood. So every single one of these contacts has the opportunity to provide regulatory information to the stem cell. And furthermore, the structure of this microenvironment is a very important component of both how information is conveyed and how cells interpret that information. So what I'd like to discuss next is that although it's very well recognized and appreciated within the biological field that biochemical cues, the identities and the presence or absence of specific molecules, can play a role in regulating the function of cells, biophysical or structural information, spatiotemporal variation in the presentation of information can be very important in how a cell interprets it. So for example, I mentioned before the extracellular matrix is in large part why we're solid phase organisms. This has not just biochemical information, but mechanical information and shape information. Furthermore, a lot of these biochemicals are immobilized onto the solid phase, and therefore there's spatiotemporal information, spatial ordering in the way that this information is presented to a cell. And if we'd like to control the cell, we need to understand how all of this information is integrated and fed into a cell. That poses a problem, however, because if I'm a worm geneticist, a C. elegans geneticist, and I'd like to understand how a cell behavior is controlled, well, I typically litter the genome of that worm with mutations, and I try to find mutations that actually change, for example, the fate of a cell, a differentiation decision. And then that provides me causal relationships between the gene sequence, the molecular property, and the cell behavior. A lot of these physical properties are not encoded by single genes. So, for example, if you take mechanical properties, these are properties of tissues that depend upon the presence of many components inside of the tissue, matrix components as well as cellular components. And to skip forward to another property, topography or shape features inside of a tissue are controlled by the identity of matrix proteins and biopolymers within that tissue, as well as the history of their processing. So as a result, it's very difficult and has been difficult within the field to develop causal relationships between complex biochemical or biophysical properties and downstream cell behaviors. These properties are difficult to manipulate genetically, they're difficult to vary, and therefore to causally establish relationships between these inputs and cell behavior. So over the past 10 years within the stem cell field, we've learned an increasing amount from the materials engineering and the polymer chemistry fields in order to effectively create materials in a process that you might term analysis by synthesis, to synthesize materials in which you can provide an x-axis in a very straightforward way to these various properties, and as a result understand how cells vary their behavior as a function of these properties, and then use this basic information to assemble functional materials that can begin to harness stem cells for downstream therapeutic application. So I'm going to, for the remainder of the talk, provide you with a couple of quick examples of advances within the field over the past 10 years in which material synthesis has been used as a tool to study biology and then can be applied as a system to mass-produce cells for downstream therapeutic application. So the first of these is mechanical properties. We're solid phase organisms, right? And in particular, I'm going to be talking about one specific mechanical property of a tissue, which is the linear elastic modulus, the extent to which a material deforms upon the application of a force in a linear fashion. So this elastic modulus has units of pascals. It can vary, obviously, over a very broad range. And we typically live within these 3 decades. So the brain, well, fat tissue is the softest tissue in the body. The brain is not that much stiffer. It varies somewhere between here and here, although the brain is very mechanically heterogeneous, it turns out. Beyond that, you get into muscle, cartilage, and bone, of course, which is a very stiff tissue. So the body ranges over a number of orders of magnitude in stiffness. So despite this richness or complexity in mechanical properties, we typically use a very blunt tool to culture the cells outside the body, plastic or glass, rock. We're taking cells out of very soft materials and putting them on top of the rock. So if you examine the extreme case of a neural stem cell, which I'll be talking about, we're taking out of this very soft tissue and putting it onto a material that's 100 million-fold stiffer in integrity. So this begs the question, does this elastic modulus affect these hallmark properties of the stem cell? And can we actually use materials, either natural polymers or else synthetic polymers, as ways to gain access to these physiologically, biologically relevant ranges of mechanical properties, and as a result begin to ask this question? So these questions were first broached within this hallmark kind of paradigm-shifting work within the field conducted by Dennis Disher and Adam Engler, initially at the University of Pennsylvania, and now Adam is at UCSD. And they took mesenchymal stem cells, which again were referred to in the previous talk, and they placed them on top of materials, synthetic materials, in which they could vary the modulus of the material. And they showed that on very, very stiff materials, the cells differentiated into bone-like cells. On intermediate stiffness materials, they turned into cells that were reminiscent of muscle, and on the very softest materials, they turned into cells that were reminiscent of being neurons. So this led to the idea that tissues have different stiffnesses, and the stiffness of any given tissue can itself serve as an instructive cue to tell a stem cell that has not yet made up its mind to turn into the cells that are characteristic of that tissue of a particular stiffness. So this left open a number of questions within the field, and in particular, we were interested in whether or not this idea could actually carry over to other tissues that are not obviously mechanically active like connective tissue is. So for example, your adult brain contains resident stem cells within this particular structure, and these have the ability, they're shown in blue, to turn into neurons to the tune of around 10,000 brand new neurons in each of your heads every day, as well as into other cell types in the central nervous system, including astrocytes. So these cells are important because they change the way that you learn and remember, and furthermore, the hippocampus is ground zero for Alzheimer's disease. So if we can learn how to control these cells, we could potentially rescue the brain from the effects of some neurodegenerative diseases. These cells can be expanded in culture and can be differentiated into the major cell types of the nervous system. And so we, as well, we're interested in understanding the effects of mechanical properties on the cells. So you can polymerize a hydrogel off of a surface. You can change its stiffness by changing the cross-link density within the material. And we call that the princess and the pea problem. How thick does the mattress have to be before the cell only feels the material? And if you put the cell on rock, on polystyrene, and then gradually soften the substrate, you can see there are fewer cells that turn into this one cell type, the astrocyte, and more that turn into neurons. And if this is quantified, on the stiffest materials, 70% of the cells are turning into astrocytes, but as you gradually soften, and this is the stiffness range of the brain where these two curves actually cross, all of a sudden you're creating greater than 90% neurons. So this is both interesting as basic information as well as useful to control cell fate. So furthermore, we've been able to elucidate some of the molecular mechanisms that are involved in this process. And a cell stiffens, it turns out, in response to the stiffness of its environment. And that cell stiffening is actually an obligate, a necessary step for the cell to be able to differentiate in response to this mechanical information in the outside world. And furthermore, once we know this pathway, it becomes druggable. We can actually control the activity of this pathway inside the brain. And for example, in this case, although I'm not going to show the raw data, by manipulating this pathway, you can actually turn up or turn down the ability of these stem cells within the brain to turn into neurons. So does this have implications for other stem cells that you may not think of as being mechanically sensitive? So as I mentioned earlier, human embryonic stem cells have been created from leftover embryos across the world. And these have the ability, for example, to turn into an important neuronal type called a dopaminergic neuron. So if a patient comes into a clinic and is presenting with Parkinsonian symptoms, they've lost 80% of these neurons already within a specific region of their brain. So it's often not enough to halt the progression of the disease. Cell replacement therapy becomes very important to consider. So you can navigate or convince a human embryonic stem cell to turn into a dopaminergic neuron by exposing it to a dynamic set of signals over time. And we simply ask the question, if you actually pulse in stiffnesses of different levels, can this influence or change the way in which this HESC turns into a dopaminergic neuron? And I'll just cut to the chase and show that if you analyze the fraction of the human embryonic stem cells that turn initially into neural stem cells, have been specified towards a neural fate, That is strongly promoted by putting them on soft materials as opposed to on hard plastic. And finally, if you analyze the fraction of the cells that differentiate into dopaminergic neurons that are expressing this marker called tyrosine hydroxylase, this can also be dramatically or significantly assisted by putting the cells on a soft material, where again, all the biochemical cues are identical between these different cases. All that's changing is the mechanical information. So I'm going to quickly end with a different property of the solid phase of the microenvironment in which a stem cell resides, which is shape features. And these include shape features on the nanometer all the way through the micron scale. And that's illustrated here that cells can be constrained and shaped by neighboring cells around them, and that shape can influence the range of things that a cell can do. And if you zoom that down, let's say by another order of magnitude so that cells are stepping on pins and needles, these shape features in the environment, and shown here is an example of what the extracellular matrix can look like once it's decellularized, this shape information is also potentially influencing how cells behave in that environment. So I'm going to take the second first. And you can use, and this has been done by a number of investigators over the years, you can use photolithography in order to introduce patterns on a surface that, for example, enable you to contrast how cells behave on a perfectly flat, not atomistically flat, but at least materials flat surface, and then on this equivalent surface, which now has shape information. And using the same tools that, of course, have been developed in Silicon Valley over decades, you can change the size of these features. And you can observe that cells are effectively anisotropic on these flat surfaces, become aligned on the other surfaces. And this influences cell differentiation. So cells tend to differentiate more into astrocytes on unaligned surfaces, and as you gradually decrease the size of the features, more of them turn into neurons. And finally, if you zoom up in order of magnitude in the shape features, this is landmark work that was conducted by Chris Chen, then at Johns Hopkins University. And he showed that if you take a mesenchymal stem cell, again, this connective tissue stem cell, put it on a very small feature so the cell doesn't have the ability to flatten out, it's spatially constrained, versus putting it onto a large feature, a 10 by 10 micron surface, where the cells have the capacity to fully adhere and spread and engage with the surface. And then you simply tell these cells differentiate into what you will. The cells on the small features ended up turning into fat, adipocytes, and the cells on the large spread out features ended up turning into osteocytes, which are cells that are involved in the formation of bone. So again, shape information, and he made some significant advances, and again, in elucidating the molecular mechanisms that were responsible for this, can strongly influence what a stem cell does, even if all the chemistry is the same. So I'd like to close by saying that material science and polymer chemistry and engineering have the ability to have an impact increasingly in stem cell biology, as it has been having an impact in biology for a number of years, where it provides a way to vary material properties in a way that you can't do genetically, to provide x-axes to processes or properties to enable mechanistic investigations of ways in which biology can instruct cell function. And furthermore, there are significant opportunities to develop new materials and to continue this process of analysis by synthesis. I'm just quickly going to provide a last example of that. Let's say you want to get from this parental cell all the way down here. Well, that environment is probably changing very, very dynamically over time. And as a result, if you'd like to simulate and control this process, you need to provide a system to the cells in which properties can change dynamically. So I'd like to acknowledge some very important work that was contributed by the organizer of this conference, Christy Anseth, in which several years ago they published a paper on a material where they ended up introducing photolabile groups into the material. So by shining a light on it, you can actually break bonds and change the mechanical properties of the material in real time in a very spatially and temporally controllable manner, simply because light is a powerful tool to pattern at that length scale and over those time scales. So when you're shining light on the material, it softens. The material is stable as soon as you turn off the light, and as a result, you can gradually program different properties into the material in a very precise way. So this is going to be a very, very useful tool in the future, I'm sure, to begin to understand how dynamic changes in a microenvironment can affect cell function. And finally, although I didn't focus on it in this talk in particular, there have been already a number of situations in which basic advances in these materials, such as the ability to mass produce a particular type of neuron, can be scalable to create scalable, controllable, reproducible, economical bioprocesses for mass producing cells of therapeutic relevance. So thanks so much for your time, and I'd be happy to answer any questions.
David. We have until 10 o'clock to ask questions.
Hi. Hello? Elizabeth Hillman. Hello? It is? Okay, sorry. Elizabeth Hillman, Columbia University. So it's curious to me. We all start out life as a single cell, right?
I'm sorry?
We all start out life as a
Yes.
single cell. And, you know, as a baby grows in the uterus, you know, all those cells have to figure out what they need to become. And all these factors that you're talking about, the mechanical environment and the chemical environment and so on, it seems there's something of a chicken and the egg argument there in terms of actual embryogenesis. And just watching what you're saying, it seems curious to me that that doesn't seem to be the case in normal sort of, like I said, embryogenesis. So I'm just wondering if there's anything else that can be learned from looking at that setting, and also to try to reconcile sort of how come these cells have these properties when maybe that isn't what influences the way that the tissues are built in the first place.
Yes, so I'll very quickly provide a comment on kind of the evolutionary transition from single cell to multicellular organisms. So at some point along the way, evolution decided that if cells can actually begin to cooperate with one another in a collective, in a very kind of communist sort of concept, that the collective ends up behaving much better or being successful as a way to copy genomes compared to the single cells. And every once in a while, an individual cell within that multicellular organism will get really selfish and decide that it wants to have its genome outrepresented, and that, of course, is cancer, right? So that is a very useful strategy, but it also provides, of course, the reason for that was that now we have, instead of a single cell type, there are around 200 different cell types within the adult organism, each of which carries out a very specific set of functions. And if we need to replace cell number 198, that means we need to understand how development created that specific cell type during embryogenesis. So what I talked about replicating in a dish is effectively the same process as goes on within an embryo. And a lot of the, for example, I show that recipe, if you will, on how to get from the embryonic stem cell to a dopaminergic neuron. The sequence of factors that were fed to the cell to do that were all learned from basic cell and developmental biology studies, typically within mouse as a model organism. So the stem cell field has embraced the idea that if you learn from developmental biology how multicellular organisms create specialization, you can do that potentially in a dish outside the body.
It's just sort of how can a brain be soft before it's a brain? See what I mean?
How can it be soft before it's, well, even within the embryo, that ball of 64 cells, the blastocyst, there's already mechanical heterogeneity. Simply due to the geometry of how cells are connected to one another, due to the matrix proteins they're already secreting, due to the cell-cell contacts. So asymmetry, if you will, in mechanical properties can be established very early on within an embryo.
Jianzhong Wu from UC Riverside. David, that's a very energetic talk. And so you emphasize a lot about the influence of mechanical properties of the stem cell. And I would imagine that the interfacial properties are probably more important. Has anyone looked into that, how the interfacial energy or interfacial tension affects the cell growth?
Well, it can affect, so there has been some work out of UCSD in which an investigator proposed the idea that the hydrophobicity of a surface can influence the cell behavior. The way it's probably doing that is by, and then there's been some work out of the Langer lab, which has actually synthesized a library of different polymers with different properties, biochemical hydrophobicity properties. And those have different influences on cell behavior. Likely, my interpretation about the ways or mechanisms by which those are functioning is that they're likely selectively grabbing different chemical components out of the solution phase and then presenting them from the solid phase in a specific fashion. So immobilization of biochemical cues onto the solid phase fundamentally changes the way in which a cell interprets that information. It sees the signal very differently in solution versus in solid phase. And so if you consider that an interfacial property, which I would, then yes, there's definitely a lot of data to indicate that interfacial properties influence cell behavior.
Yeah, I understand that everything is related, but consider that the two surfaces with the same mechanical properties, but different interfacial tensions.
Oh, sure.
And I would imagine that the cell growth that the growth rate might be very different?
They would. I mean, each one of the properties I kind of showed on that slide, as well as the concentrations of a number of biochemical factors, each one of those represents an axis, right? And this is highly, highly multidimensional space. So, and they're probably very nonlinear interactions between the different axes. The stage of the field right now is that, especially with the solid phase, now we understand that these particular material or biophysical properties are important individually. You know, we've gotten an x-axis. We can vary it. And the next set of questions, of course, is going to be understanding how these combinatorially affect functions of cells, and that provides important mechanistic information as well as teaches us how to create a culture system to mass produce the cells.
Kevin Turner, University of Pennsylvania. So sort of phenomenal control over the cell behavior is very impressive. And if you gain this ability to control the cell fate and differentiate into whatever type you want, what's the end goal? So how does this scale to sort of an engineering solution? Is the goal to produce many types of one cell and then inject them to the body? I don't come from a bioengineering field,
Sure.
so I just want to sort of see what's next.
Likely the lowest hanging fruit, if there is such a thing, are going to be diseases in which a single cell type can be injected in an amorphous fashion from a stem cell. I think that there's, as we saw in the previous talk, there's in parallel a lot of advances going on in creating structure in the cells that would be implanted. Within the stem cell field, things like, well, Parkinson's disease, where implantation of a dopaminergic neuron, a single cell type, can have an impact. It's been shown to have an impact in some clinical trials. Age-related macular degeneration, where you're trying to rebuild the barrier between the vasculature and the retina by implanting the cell called a retinal pigment epithelial cell. That's in clinical trials right now for RPE cells derived from human embryonic stem cells. Type 1 diabetes, where the cell type that's missing is a beta cell because the immune system made a mistake and killed off that beta cell, and therefore the source of insulin within kids who have autoimmunity against their beta cells. That's a single cell type that has the ability to create insulin. So those are really the targets the field is shooting for right now, and in many cases, there's no way to create those cell types in industrial scale. So we need to learn much more about all the varieties of signals that regulate the differentiation processes so that we can implement that, I believe, in materials that are cheap, scalable, synthetic, reproducible, safe.
Reed Hendershot, Air Products. I also do not come from a biological background, so maybe it's a simple question, but the embryos that are donated by couples, are those usable just for research, or can they be made into something and put into someone's body, or will that body reject those because they're not used? So when you're doing therapy, do you have to get stem cells from a person, the same person they go back into, I guess?
Sure. So the first question is that, typically for IVF, around 8 embryos, 7, 8 are created, are fertilized in vitro. And then typically something on the order of 3 to 5 are implanted. That's why there are lots of identical twins and triplets for IVF. The remainders are frozen down in case the first try didn't work out. If the parents are lucky enough, that's where, to have kids the first time, that's why all those leftovers are still around. And right now, nobody knows what to do with them. There hasn't been a program to systematically bleach them, which is what would happen probably. And as a result, there's been kind of a slow trickling of them for scientific use. One positive property of the stem cells is that they have this ability for self-renewal. They can indefinitely expand, so you can start out with a single cell and make Avogadro's numbers of them. So as a result, you don't need to be deriving new stem cells every time you'd like to do an experiment with the human embryonic stem cells. There's some standard lines that most everybody in the US and around the world have. A lot of them come from Wisconsin, for example. As for the tissue compatibility issue, in some countries, the major histocompatibility loci, the genes that are responsible for kind of tissue identity and tissue rejection, that genetic pool is somewhat limited. So for example, in Japan, there's a relatively narrow range of MHC alleles. And as a result, probably something on the order of 100 different lines could be banked and provide histocompatibility, tissue donor compatibility, stem cell donor compatibility with the majority of the Japanese population. For the rest of the world, it's complex. We have very, very heterogeneous histocompatibility pools, and that's still an open problem. So it's probably going to be some combination of matching the compatibility of the stem cell or the cell derived from the stem cell to an individual recipient, as well as immunosuppression, same thing you would do if you got a liver transplant.
Jason May, HRL Labs, and again, I'm a non-biologist, so please pardon my ignorance, but I was wondering how the cells sense the stiffness, and if that can be replicated or simulated by vibration or compression in a centrifuge or other methods.
So that's a really complex question. Probably the stiffness sensor is going to be almost a continuum of molecules from the outside of the cell all the way to the cytoskeleton, this network of proteins on the inside of the cell. There's some people argue, like Viola Vogel, for example, argues that if you put a force on an extracellular matrix molecule, this big molecule on the inside of the cell, you can actually pull it apart, unfold it, and begin to expose new biochemical information that the cell can sense, can bind to. So in that case, the idea is the mechanosensor is not even inside the cell, it's on the outside of the cell. Some people believe the mechanosensor occurs at the level of the receptor, that once these receptors on the cell surface, these adhesion receptors, integrins, bind to the matrix, they can be spatially patterned, can aggregate, create clusters, and it's that process that converts the mechanical information into biochemical information. And furthermore, the people who believe that the assembly of proteins onto these integrins, a variety of focal adhesion proteins, as they're called, serves as the mechanosensor. Probably all of those are true. So it's a very complex process. And can you simulate that? Well, all of the tissues in your body receive a 1 Hertz strain, right, due to your heartbeat. The brain does as well. So I showed to you sort of static mechanical properties, cyclic mechanical properties, or cyclic strain is a whole different type of mechanical cue that a cell can receive. And there's been a lot of work, which I did not discuss at all, on how that affects cell behavior and stem cell behavior.
Andrea Armani, University of Southern California. So I have a related question. So you showed 2 results from 2 different labs. One where it was a small circle and they formed one type of cell, and then related experiment was a large square and it formed a different type. And then the other set of experiments, it was a flat wafer and it was randomly oriented, and then they patterned little lines and everything all lined up nice and pretty. Did they ever compare those 2 results? Because it would seem like the confinement in the lines would have encouraged the change in the cell type, kind of like the confinement in the little tiny circle would have changed the cell type.
Sure. So as you kind of scale down, let's say from patterns are on the order of 10 microns and then get smaller and smaller and smaller, and then lithography can get you to, of order 100 nanometers or so. Probably, that's an x-axis. That's a valuable x-axis to be able to vary. The next question is how are cells mechanistically sensing that information? It's probably through a variety of things, changing the structure of the adhesions between the cell and the surface, changing the alignment of cells as they end up squeezing into the grooves, changing the frequency with which cells come into contact with one another. So right now, I think that field is at the stage of they have a phenomenon, and then the next question is to hypothesize mechanism and be able to eliminate or narrow in on those various possibilities as the underlying mechanism. But right now, we really don't know.
Steve Little, University of Pittsburgh. So it's interesting you're talking about axes and making materials that have a certain axes, and then you can measure how that material affects the fate of the cell, differentiation state of the cell, activation state of the cell. But in the field of immunology, you have some signals, like let's say a biochemical signal, and you can have more and more of it and see one response. But then perhaps patterning, which is a different axis, interacts with that axis, and then you actually don't need a lot. You need a little in a pattern. Or another example of that would be that you could have a certain number of biochemical signals, but if you change the order or presence, you get a completely different stimulation or a completely different differentiation state. Is that possible in this, and could we be potentially moving forward need to be very careful in the conclusions that are made because of the unknowns in the different number of axes of information in that?
Oh, I fully agree with you. We actually have a body of work maybe we can talk about. You're really saying that biology codes molecular information in more than just a binary signal. Is molecule there or is molecule not there? There's a lot more richness than that, right? It's how much and where is a function of time. And there's a large number of molecules that, I referred to spatial immobilization of molecules, which I think we may be talking about the same thing there, that if you spatially immobilize something, then all of a sudden, if a molecule is diffusing on the outside of the cell, every direction the cell sees the same concentration. But if you can immobilize that molecule, you can spatially pattern the way in which it's presented to the cell. And as a result, the cell ends up seeing a very, very different set of information. So you can take the same molecule and A monomer would signal one way, a dimer would signal another way. If you create an oligomer of, let's say, 20, if you have the same number of molecules, but all of a sudden your 20 are spatially constrained, the cell ends up, as you're suggesting, experiencing a very potently different response. So that's been known for many years in immunology, and the stem cell field is learning now from immunology, that spatial patterning in the presentation of cues can have a very dramatic effect.
Yeah, thanks for this very interesting talk. I'm also outside of this field of biology. So one quick question about the modulating the cells. What would be the frequency that you are using? Also, different cells react differently in terms of the frequency range. And also, when you talk about the system to be nonlinear, what do you refer to? Is it nonlinear to power or to some other parameter?
So the first part, when you're talking about frequency, are you talking about strain or
the light, the optical?
Oh, light.
Yeah.
So frequency of light introduction into the material.
Mm-hmm.
So I may defer to Christy to that. If you're talking about using light to be able to break bonds inside of a material and change the properties of the material. Well, I mean, those the photolabile groups that she utilized, absorb light within a particular wavelength of the spectrum or range of the spectrum.
So you need to be.
So you would, presumably pick the peak of that or near the peak of that to be able to break the bonds most effectively. And then there's an idea that potentially you could actually introduce multiple photolabile groups that respond to different parts of the spectrum and use that to manipulate different properties inside the material and in orthogonal ways.
Tweezer, yeah.
And so then what was the last
as a
question?
Nonlinear property.
Nonlinear property within a material, for example?
Yeah, so what do you refer to?
So a nonlinear mechanical property would be, for example, strain stiffening or strain softening.
Okay.
That if I push on my hand, there's a linear proportionality between the amount of force and the amount of deformation.
Okay.
In some cases, the more you strain, the stiffer the material becomes. And it's been proposed by a guy named Paul Janme that can provide a way for cells to communicate with each other through a material. That if one cell pulls locally and it stiffens the environment around the neighborhood, that's a mechanism for cell-cell communication.
Thank you.
Two last questions, one on this side, if you go ahead.
Hi, for Vanderbilt. So you talked about the human embryonic stem cells. Why, since 2008, aren't we hearing more about the IPS? Because can you give a differentiation and the success of those?
Yes. So in 2006, a guy named Shinya Yamanaka, who will win the Nobel Prize one day, it's just a matter of time, found this idea that, when Ian Wilmot first created Dolly the sheep, the way he did this was he took an adult mammary nucleus and stuck it into an egg, at the cytosol of an egg. And some proteins within that ball of biochemicals in the egg entered into the genome, into the nucleus of that adult nucleus, and rebooted it, tricked it into thinking it was an embryo again. So turn back the clock on that DNA, and if you turn forward the clock again, you create an organism. That's how Dolly was cloned. So for a number of years, investigators were wondering exactly which proteins is it within that ball of molecules that's responsible for rebooting the genome. People thought it was going to be too difficult to narrow down from, let's say, 10,000 proteins to just a couple. So Yamanaka discovered all you need is 4. So if you overproduce or introduce 4 proteins into the nucleus of almost any adult cell, you can reboot it and churn it into an embryonic-like cell. That's an induced pluripotent stem cell. So that's a major advance within the field, and that leads to the consideration you can get pluripotent stem cells that can create everything in the body without actually destroying an embryo. And given the choice, of course, you would rather not destroy it. So some of the results I showed you, for example, the mechanosensitivity of human embryonic stem cells, we showed that applies to human-induced pluripotent stem cells as well. That field is incredibly active, and I was in Japan just a couple months ago, and they've completely shifted over from studying human embryonic stem cells and are almost 100% iPS cells now.
Thank you. Very nice talk. Thank you for making it accessible for a lot of us. So my question actually is, I'd like to hear your opinion about it, and also maybe see the question for before the break that maybe people can discuss. So what is our end goal? You say basically, as we make progress in medicine and these therapeutic methods, we better the quality of life and maybe lengthen the life of the individual. So do you think we can get to a point where, what is the end goal? Do we want to make everybody live forever? Or what are the implications on a global scale where let's say 100 or 200 years from now, or I don't know how long, if we achieve everything that we set out to achieve in terms of turning off cancers, creating regeneration, where will we end up and do?
Sure. That's a very philosophical question. I'll try to do my best to address, and I'll also kind of inject some, economics into it. So probably at some point there's going to be a finite, length of a life where multiple organs are going to fail and somebody's time has come. And what we're interested in doing is identifying situations where people prematurely experience diseases or conditions that rob them of quality of life and productive years. Parkinson's is a really good example. It hits in the 50s. When somebody's in their prime, their productive prime, they're still parents. Their kids still need them, increasingly so these days. And in addition, it robs them of their absolute peak in contribution to the economic well-being of the country, right? Alzheimer's, you can make the argument, well, that typically hits later in life. But for every Alzheimer's patient, there's somebody who has to be employed full-time to care for them. That's very expensive as well. So up until somebody's time has come, where our goals are, our interests are in identifying costly diseases, costly both in terms of personal quality of life as well as an economy for the entire country, and making them cheaper to treat.
Great. Thank you so much.